Hello everyone, welcome back to the class of biotechnology. Today we are going to discuss about the cell cycle. Cell cycle divided into three phases. That is the interphase, mitosis and cytokinesis. But before moving forward, if you are new here, please like and subscribe this channel and don't forget to hit the notification bell so that you cannot miss any of the future video. One of the greatest wonder of science is the mechanism by which a single cell, the result of the fusion of the egg and sperm, develops into a complex multicellular organism such as human. You know, this single cell, it is the resultant of what? It is the resultant of egg and sperm. When egg and sperm fuse together, they form a single cell. And this single cell is called as zygote. You know, all these zygotes undergo mitosis in all situations, meaning there is no exception. So when a single cell divides into its daughter cell, then that daughter cell further divide into their own daughter cells, and in this way, by following the process of the cell cycle, a trillions of cells can be produced. And when these trillions of cells are put together, they form a complex multicellular organism. This multicellular organism is us human beings. So we are the example of eukaryotic multicellular organism. In this way, cell produces more cells by the process known as cell cycle. Cell cycle are further divided into three phases, that is the interphase, mitosis and cytokinesis. Let us discuss about the interphase. Interphase is also called as preparative phase because in this phase cells prepare itself for growth and division. But actually in this phase cells only grow in size. They do not divide in this phase but prepare itself for the division. Before going to the stage of mitosis, cell prepare itself for the division by producing the required organelles. This interphase is further separated into three phases, that is the G1 phase, that stands for G actually stands for gap, and in this phase cells grow in size. Next, in S phase, the cells allow the genetic material, that is the DNA, to replicate and synthesize. In this phase, a single DNA molecule unwinds itself and produces its daughter DNA. In this way, cells allow the process of replication. That's why this phase is called the synthesis phase. S here stands for synthesis. Next, when moving towards the further G2 phase, cells is now almost prepared for the mitosis. Here, the cells again grow in size and allowing all the organelles to get prepared for the process of mitosis. In G2 phase, cells such as muscle cells and brain cells do not undergo further division after reaching to the G2 phase and remains in zero phase. Hence, the muscle cells and brain cells are said to exist in the zero phase. This zero phase is represented by G0. So this is the major event of the interphase. Like in this interphase, you will be seeing about the centrosomes, that is the two pair of the centrosomes. In this phase, you can visualize the plasma membrane, chromatin. The genetic material here resides in the form of chromatin. And then the nuclear envelope you can see in the interphase and nucleolus. Now, the cell in the interphase is having a distinct nuclear envelope and the genetic information is dispersed in the nucleus as chromatin, right? Now the cell is fully ready to undergo the event of mitosis. Mitosis is a continuous event and is divided into four distinct phases, that is the prophase, metaphase, anaphase, and telophase. In mitosis, the chromatin in the nucleus undergo the condensation and get compact and form a chromosomes. The chromosomes consist of two chromatids. One is this one and another is this one. And these chromatids are held at the center by the centromere. In the prophase of the mitosis, the chromatin starts condensing and the chromatin get condensed into the chromosomes. You can see here the condensed chromosomes. You know that the chromosomes consist of the two chromatids, and these two chromatids are attached at the center by centromere. So this is the centromere. This portion is the centromere and these are the two chromatids, right? In prophase, the spindle fiber emerges from the centrosomes. This portion is the centrosomes, and from the centrosomes you can see spider leg-like structure. So this is called as the spindle fiber, right? This is the centrosomes and this spider leg-like structure is called the spindle fiber. In the prophase, the nuclear envelope breaks down and it starts disappearing. Nucleolus disappears, right? Nuclear envelope starts disappearing by breaking into different fragments. It started disappearing. And in the late prophase, or you can say in the early pro-metaphase or early metaphase, the chromosomes continue to condense. And here, the kinetochore appear at the centromere. Here at the centromere, kinetochore starts appearing. And the mitotic spindle, microtubules attached to the kinetochores. This spindle fiber, you can call it as a microtubule, mitotic spindle microtubule. You can call it as a mitotic spindle microtubule. So this gets start attaching to the kinetochore like this here, you can see. And in this phase, the centrosomes start moving towards the opposite pole. So you can see here this centrosomes is allocating towards the pole. Here, as soon as the centrosomes start moving towards the opposite pole, the cells entered into the metaphase. In metaphase, the mitotic spindle, that is the spindle fiber that is originating from the centrosomes, are fully developed, right? Are fully developed. As you can see here, it is fully developed, right? And the chromosomes lined up at the metaphase plate here at the equator. And these spindle fiber that is originating from the centrosomes are attached to the kinetochore of the centromere. Here, each sister chromatid is attached to the spindle fiber originating from the opposite pole. As I have said before, the spindle fiber that is originating from the centrosomes from the opposite pole is attached at the kinetochore of the centromere, and this centromere is holding what? Is holding the sister chromatid together. After this event, the cell undergo into the phase of anaphase. In anaphase, the chromatids separate at the centromere, and each chromatid is now known as daughter chromatid. You know that the sister chromatids are held together by the centromere. In anaphase, these centromere are breakdown due to the breakdown of the cohesion protein, and due to this breakage, this sister chromatids that were held together by the centromere is also separated out, and now this is called as daughter chromosomes. This is now called as daughter chromosomes. The spindle fiber attached to this region, right? Attached to this region. This region is what? This region is centromere. The spindle fiber that is attached to the centromere pulls the daughter chromosomes towards the opposite pole, towards the centrosome that is present at the opposite pole. The centromere region of the chromosomes move first, as you can see here, and the arm of the chromosomes follows. Now in telophase, when the daughter chromosomes reach towards the pole, the telophase begins. The daughter chromosomes begin to unwind into chromatin. Here you can see that it is getting unwinding, uncondensing. The nucleolus reappear, you can see here, and the nuclear envelope begins to reform. You can see here the nuclear envelope. Nuclear envelope. The mitotic apparatus disassemble, thus terminating the mitosis. Here, a furo-like structure start appearing. In the stage of the cytokinesis, the splitting of the cells cytoplasm into two parts is known as cytokinesis. Here, this is the cytoplasm, and here is the another cytoplasm of other cell. Although cytokinesis is a distinct process, it frequently begins during the late anaphase or early telophase. In late anaphase, as the chromosomes are moving towards the pole, the plasma membrane begin to constrict at a region known as cleavage furo. So these two regions, these are the cleavage furo. Cleavage furo. And this begins the process of dividing the cytoplasm, ending as the cell splits into two daughter cells. The cytoplasm and the organelles are effectively divided into two parts here, as you can see. In plant cells, a cell plate separates the daughter cell. But in animal, there is no cell plate present. Only the cleavage furo separates the daughter cells. Right? So thank you so much for today. I hope you like this video.